This two-part series begins with a focus on the molecular fluid shift, specifically the distinct biochemical mechanisms of secretory and osmotic diarrhea. Video 1b will address the resulting tissue damage and anatomical localization. To identify the origin of these shifts, we have to strip away the gross anatomy, moving past the immune responses and mechanical peristalsis, down to the cellular scale, stopping at the isolated lipid bilayer that divides the intracellular environment from the intestinal lumen. At this boundary, we have to look for the exact cellular mechanics that force water across this semi-permeable barrier against the body's homeostatic drive. In a regulated state, the epithelial barrier maintains a precise balance of ion exchange and water transport to keep the body hydrated. Pathological fluid loss occurs when this balance is disrupted by two distinct modes of failure: active enzyme-driven cascades within the cell, or the passive physical chemistry of the lumen. Understanding luminal fluid accumulation requires viewing it as a failure of either signal transduction or thermodynamics at the membrane level. Secretory diarrhea is an active transcellular process where the cell's internal transport machinery is hijacked by pathological signal transduction. The process begins when an exogenous toxin, such as those produced by cholera or E. coli, finally arrives at the apical membrane. This toxin targets the GS-alpha subunit of the heterotrimeric G protein complex. Under normal conditions, GS-alpha has an intrinsic GTPase activity. This functions as a built-in off switch, hydrolyzing active GTP into inactive GDP to keep the signal brief and controlled. The toxin interrupts this cycle by catalyzing the ADP ribosylation of the GS-alpha subunit, binding an ADP ribose molecule directly to the protein. This specific molecular lesion destroys the protein's ability to hydrolyze GTP. Because the cell has lost its regulatory off switch, the GS-alpha subunit remains locked in a permanent constitutively active state. The locked GS-alpha subunit provides unrelenting stimulation to an adjacent membrane enzyme, adenylate cyclase. Stimulated adenylate cyclase rapidly converts available ATP into cyclic AMP, flooding the interior of the cell. Without a way to turn the engine off, the concentration of intracellular cyclic AMP begins to spike exponentially. This high concentration of cyclic AMP binds to and activates protein kinase A, or PKA. A single toxin interaction at the surface has now been amplified into a massive intracellular signal through this secondary messenger cascade. Once activated, PKA migrates back toward the apical membrane. Here, PKA phosphorylates the R domain of the CFTR chloride channel. This phosphorylation structurally locks the CFTR channel in an open conducting state. Chloride ions begin pouring out of the cell and into the lumen. This purge of chloride creates a massive electrochemical gradient. To maintain electroneutrality, sodium ions are forced out into the lumen to follow the chloride. Water then follows the sodium via osmotic draw, meaning the cell is effectively pumping itself dry. Clinical Pearl for the audience. Please like and subscribe for more custom medical videos made just for you. Now, moving on to the next segment. In secretory diarrhea, the cell's own internal transport machinery is weaponized against its fluid balance through biochemical hijacking. We now shift our focus entirely to the fluid-filled expanse of the luminal space. In the osmotic mechanism, the cell's internal machinery is functioning perfectly. The pathology here is driven by entirely luminal physical chemistry. This occurs when poorly absorbable, osmotically active solutes, like magnesium or undigested carbohydrates, enter the gut. Because these solutes are chemically incapable of crossing the lipid bilayer, they accumulate in the lumen. The buildup of these solutes raises the osmolarity of the luminal fluid, setting a thermodynamic trap for the body's water. The shift is driven by a simple physical principle. Nature acts to eliminate an osmotic gradient. The high concentration of trapped solutes exerts a continuous osmotic pressure against the epithelial barrier. This force pulls water molecules out of the intracellular space and into the lumen. The water is held in the lumen to dilute the solutes until isotonicity is achieved. In osmotic diarrhea, the transport channels are simply overwhelmed by the thermodynamic force of the trapped luminal particles. These two distinct molecular environments produce the same result, an accumulation of fluid in the lumen. Because the secretory mechanism on the left is driven by a locked intracellular signal, the fluid loss continues even if the patient stops eating. The osmotic mechanism on the right is purely passive and depends entirely on the physical presence of the offending solute. If the intake of that specific solute stops, the osmotic gap collapses and the water retention ceases. Identifying these specific mechanics, the aberrant G protein signaling cascade versus the luminal thermodynamic trap, explains the molecular basis for two of the primary drivers of intestinal fluid shifts. Up to this point, our focus has been on functional transport. We have examined pathologies where an intact, highly selective epithelial filter makes mathematical errors, secreting too much chloride or failing to absorb enough sodium. But inflammatory diarrhea operates by an entirely different mechanism. It is a physical collapse of the epithelial wall itself. Look at this cross-section of a healthy mucosal barrier. The perfectly aligned row of enterocytes forms a continuous seal that isolates the sterile, nutrient-rich bloodstream below from the pathogen-heavy environment of the lumen. We are going to track exactly how microscopic molecular signals dismantle this physical wall, and how the geometry of that destruction dictates the symptoms observed at the bedside. To accurately diagnose the source of a patient's gastrointestinal distress, we must move beyond functional models of fluid transport and analyze the physical architecture of gut tissue destruction. A functional transport error produces clear, watery output. Inflammatory diarrhea produces an exudate. This specific output is the primary clinical indicator of structural damage. With the epithelium degraded, the lumen collects a complex mixture of internal fluids, departing from the water loss seen in osmotic transport errors. In this diagram of a breached mucosa, observe what escapes. Whole blood, plasma proteins, and purulent mucus spill from the basolateral capillary beds through gaps in the lining. This accumulation in the lumen defines an exudate, presenting clinically as dysentery. This leak is not a passive process of degradation. The body's own immune system actively engineers this tissue damage to clear an infection. Dysentery serves as the macroscopic evidence that an active inflammatory process has physically shattered the microscopic barrier. The destruction begins in the lamina propria. Here, a resident macrophage detects an invasive bacterial pathogen by making physical contact with its surface antigens. The moment detection occurs, the macrophage releases a specific cascade of molecular signals called cytokines. The first specific driver in this cascade is interleukin-8, or IL-8. IL-8 establishes a potent chemotactic gradient. The macrophage releases the cytokine in concentric waves that diffuse toward the local capillary bed. This gradient acts as a homing beacon. When IL-8 waves intersect the capillary, they signal circulating neutrophils to adhere to the endothelial wall. Massive recruitment follows. The neutrophils deform their cell bodies, actively squeezing through the tight gaps in the capillary endothelium in a process known as extravasation. Thousands of these neutrophils pull themselves out of the bloodstream and flood into the lamina propria and the mucosal epithelium, preparing to engage the pathogen. IL-8 acts as the logistical signal that converts a stable mucosal layer into a staging ground for a destructive cellular immune response. While IL-8 recruits the inflammatory cells, a parallel cytokine pathway compromises the architectural integrity of the epithelial wall. This pathway is driven by tumor necrosis factor alpha, or TNF-alpha. TNF-alpha targets the tight junction complexes. These two adjacent enterocyte walls are held together by interlocking protein blocks known as claudins and occludins. TNF-alpha forces the downregulation of these proteins. As claudins and occludins dissolve, the cell walls drift apart, opening a wide physical channel. Unzipping the tight junctions is only the first phase of the damage. TNF-alpha also initiates direct enterocyte apoptosis. This triggers programmed cell death. The intestinal lining cells collapse and fragment, leaving physical microscopic craters throughout the mucosa. The combination of open paracellular channels and missing cells transforms the barrier from a selective filter into a permeable sieve. By dismantling the physical wall, TNF-alpha ensures that the ensuing immune battle will spill blood and cellular debris directly into the intestinal lumen. Once this microscopic tissue destruction occurs, the patient's clinical presentation is governed by gross anatomy. The anatomical localization principle states that the physiological capacity of the organ and its embryological innervation dictate the symptoms. Let's apply this to the small bowel, the jejunum and ileum. As this grid illustrates, the primary physiological trait of the small bowel is its massive absorptive surface area. When inflammatory damage strikes an organ of this capacity, it impairs absorption across a massive surface, resulting in large volume fluid loss. To understand the patient's pain, we look at the embryological origin of the small intestine, which derives from the midgut. The visceral pain fibers for midgut structures run alongside the lesser splanchnic nerve, terminating exactly at the T10 level of the spinal cord. Because the signal enters the T10 dermatome, the brain interprets this as diffuse periumbilical pain, a deep ache centered around the umbilicus. High-volume fluid loss paired with periumbilical pain provides a diagnostic signature for small bowel inflammation. The large bowel, comprising the colon and rectum, serves a different physiological role. The colon operates as a low-volume desiccation and storage organ, not a primary fluid processor. Consequently, inflammatory barrier collapse here produces significantly less total fluid loss compared to the small bowel. While the output volume is low, the pain presentation is severe, driven by the specialized anatomy of the distal colon and the rectal vault. This cross-section shows the specialized stretch receptors embedded into the muscular walls of the rectum. The local cytokine storm makes these receptors hypersensitive. They fire intense electrical signals into the pelvis, even when only tiny amounts of inflammatory exudate enter the vault. The result of this hypersensitivity is tenesmus, the continuous, painful, and often unproductive urge to defecate. The pelvic splanchnic nerves carry this localized signal, driving the clinical symptom of high-frequency, small-volume urgency. Small-volume output paired with agonizing tenesmus confirms that the inflammatory pathology is localized to the large bowel. The cytokines IL-8 and TNF-alpha scisipally destroy the epithelial barrier, generating a blood-rich exudate. The clinician can confirm the location of this tissue damage by measuring output volume and identifying the location of the pain. The diagnostic patterns are consistent. High volume with periumbilical T10 pain identifies the small bowel. Low volume with tenesmus identifies the large bowel. The patient's gross symptoms serve as a direct map to the microscopic site of cellular destruction. Understanding anatomical localization explains the clinical presentation through the mechanics of the tissue itself.